Picking up chapter 3, a couple of pertinent things. Remember, this is what drives people crazy about science. Sometimes what happens is you think the two reports are contradictory, and they aren't completely contradictory, like the effect of low sodium diets, where one study found no effect and one study found a statistically significant effect. Well, one of them was looking at recurrent heart attacks in people that had had heart attacks, and those did drop compared to the control group. And the other group looked at overall mortality in people that had heart attacks. And overall mortality compared to the control group was the same. They weren't dying as much of heart attacks, but they were still dying at about the same rate. So they look contradictory, and you look at that and go, well, something odd's going on, and maybe we can figure that out, right? We're always trying to explain things. But they weren't really opposed to one another. They weren't really contradicting each other. But that happens. You have to kind of look kind of closely sometimes at results. Hopefully you don't run into this. Since people do these things, there are times where peer review feels like this, where it just says, that's it, that's peer review. If you have a new and kind of revolutionary idea, then you may get a lot of opposition from people that have been around for a while and are not really prepared for a big revolution in how they see the world. And so some ideas are just harder to get out there, to get them published, to get them high profile, and they can take a while to work their way into the lexicon, as they say. So now we're going to talk about the process. The caption here says, you may be right about the house tilting. She has a hypothesis based on an observation. He makes an observation and goes, oh, wow, I'm seeing evidence to support your hypothesis. The process starts off with observations. This is called scientific method. And it starts with observations. Police, a lot of dead cats. The caption says, notice all the computations, theoretical scribblings, and lab equipment, Norm. Yes, curiosity killed these cats. Now that's a bit of a leap, hypothesis-wise, but he's telling you the observations that he's making and that he's leaping to an explanation. That's how this works. You start with an observation, and that leads you to an explanation, a hypothesis. Well, it turns out there's a few ways to do this. The cartoons are direct observations. They're taking their observations in directly through their own senses. Indirect observations, you get secondhand. It can be through somebody else's direct observation. You read stuff, that's indirect observations. You look at somebody's conclusions and go, oh, I think something else is going on here. You look at their results, you go, oh, those were interesting results. I don't like the way they interpreted them. I would interpret them differently. Is that all sorts of secondhand stuff is one type of indirect observation. The other type of indirect observation involves things that we want to know about the world, but our own senses can't tell us. We want to know about very, very tiny things, but we can't see very, very tiny things without a microscope. We can't see distant things in space without a telescope. We can't listen to bat sonar without an oscilloscope that can record frequencies of sound that are beyond our ability to detect. So the second type of indirect observation involves when we use technology to supply us with information we can't get ourselves. And again, so it's secondhand, And the same way that secondhand from other people, you're trusting that their observations are really what was going on, the same thing happens with devices where sometimes you'll see something and it turns out that the device is messed up. It's telling you something that isn't really there. You have to figure that out. So secondhand always has some weaknesses to it, even though direct observations have some weakness to them as well. So you come to your explanation, your hypothesis. This is what I think is going on. There's two features a hypothesis absolutely needs to have that lead into the basic structure of the science system. Got to be testable. And testability is based on if-then structure. Notice that doesn't say predictable, it says predictive. Those are two different words. But how do you design a test? If I'm right about this, then if I do this, this should happen. You're making a prediction based on your idea. Your idea is predictive. Is that Einstein said, if I'm right, then during a solar eclipse, the bending of space-time around the sun should change the position, the apparent position of the stars. And that was a test that nobody could do for quite a while because they needed to be set up on a solar eclipse to do it. But if you can't go, if this, then that, how do you design a test in the first place? So this is absolutely a necessary ingredient in order to get to this. Your idea has got to be predictive or it isn't testable. Now it'd be nice if we could do highly organized classic experiments every time we have an idea. But we can't, because we don't have a whole bunch of things. Is that there's a whole bunch of predictions that we need to make about the world that require us going out into the world to test them. Maybe that's the only way that you can test things. When we go through the lab, we'll run into situations where field test is really the only logical approach. But you can't really structure an experiment perfectly when you run a field test. But field tests could be, you can't really duplicate natural conditions in your organized lab. You can't afford to do it. It isn't ethical to do it. You can't be pulling endangered species out of their natural environment in order to run some sort of test on them in the lab. It may be ethical to go out and do stuff with them out in nature, but there's all sorts of parameters that say you got to go out into the world to run this test. And there's got to be limitations into how you do that. And sometimes you run into a test and you go, why are they even testing this? What the heck does this even mean? The caption says, testing whether or not animals kiss. And he's kissing what appears to be an aardvark, and the cow is popping him in the head. And on the blackboard it says, armadillo, looks like rejection, doesn't. I don't know what it says there. Squid, couldn't find lips, no response. Something about latest study. But there are times where you see tests and you go, I don't really understand where they even were going with this. And sometimes the study is, people go, well, I know what you're gonna find. It's really obvious. We all know how this works. And sometimes those are the most interesting studies because people test something that everybody accepted and it turns out not to really work that way. It's a classic experiment about crack babies, is that women come into the hospital, they've got high levels of cocaine in their system, they've been addicted to cocaine through their pregnancies, and people go, well, yeah, those babies are messed up. And we can tell you exactly how they're messed up. And there was a researcher at Johns Hopkins who wanted to do some follow-up studies and then looked to see what the original studies had shown, and found out there were no original studies. So he decided he was gonna do like a basic study. Is there a real effect here that is a recognizable effect? And because he had access to Maryland, in Baltimore, cocaine, major problem at the time, he set up a study. They had a whole bunch of what they call neonate units, what we call nurseries, where they send the babies after the babies have been born. And so he selected a couple of them. And what normally would happen is that the babies born to mothers with cocaine in their system, or mothers who were on record as having cocaine problems, that would be on the chart, and it would go up to the neonate unit with the babies. He went to the neonate units and he said, we are not going to include that in the charts. But what we're gonna try to do is send you roughly 50% babies that would be designated crack babies and 50% babies that would not be designated crack babies. And we're gonna hang on to them longer than normal. We're gonna hang on to them for a week. Normally we send them back out as fast as possible. But all we need you to do is at the end of the week, tell us which ones are which. And the nurses went, this is stupid. They said, there's no problem, it's gonna be easy. And as a control, they took each baby and flipped a coin as a predictor. Crack baby heads, non-crack baby tails. Now, the coins didn't know. Well, it turns out the nurses were no better at matching the babies with their designation than the coin was. Is that without the input from the chart, the behaviors that they saw weren't significantly different, but they were sure they were going to be fussier and smaller, and they wouldn't develop as well. Is that the nurses were sure they were going to see effects that everybody accepted were effects in crack babies, but without the designation, they couldn't really see that. Is that, and you learn that your own biases tend to produce a narrative in the world that confirms your biases. That's what happened in this situation. And other kinds of studies have found out, from a biological standpoint, it makes absolute sense that cocaine during the course of a pregnancy would affect the brain of the infants and affect a lot of stuff. But it turns out, if you do a very neutral experiment like that, where it's like, okay, let's really look for the differences, and ignore and try to factor in the confounding factors. We'll talk about those eventually. It turns out they couldn't tell the difference, which kind of threw all sorts of monkey wrenches into the works of some studies that had been follow-up studies to the assumption that there was a difference. So now we're talking about design. When we do the experimental design lab, is that one of the things that I say over and over and over again is, okay, here's the setup. What's the variable here? And what's the thing that changes? Well, that's what the word means, but that's not the function of a variable in experimental design. Experimental design is all about comparisons, like Reddy's meat, and covered meat and non-covered meat. And the idea is that varies between your two tests. And ultimately, if you can do it, it's the only thing that should vary between your two tests. So that if something happens over here that doesn't happen over here, you can connect that to your variable. Your variable is what you're testing. And so the first thing you have to decide is, okay, what exactly am I gonna be testing here? And your best designs are very focused on a single thing. If you got 3 different things in your experimental design that are different between your two tests, and you get differences, well, which one of the 3 things produced the differences? Were they all equally involved? Was one of them not involved and the other? Is that you want to focus. You want to pick one thing if you can and focus your study design on that. And so that's the variable. In the case of the crack baby study. The variable was some babies would have been designated on paper crack babies, some babies would not have been designated crack babies. The data you were collecting was, can trained professionals who work with infants all the time tell the difference without any kind of input other than what's in front of them with the babies? And so it was very simple data. Are they or aren't they? Is it in the cases of, like right now, they're trying to figure out how much immunity exposure to COVID gives you. And people are they're measuring antibody levels, because that's a simple thing to measure. But a lot of people who are trained in immunology are saying that's not a great way to collect your data. It doesn't really tell you. You can look at the people with, with COVID and people that didn't have COVID, or look at their antibodies early and their antibodies late. And the change in levels isn't as important as these other ways you would better measure immunity. So going into a design, it's like, what are we trying to find out? And how do we measure it in a way that really answers the question? When you're designing a vaccine, how do you know whether the vaccine is effective? Yeah, we can leave them for years and see if they get sick again. But to get the vaccine approved early, we need to measure measurements of immunity to a particular input that's going to tell us whether the vaccine is working. And so they look at antibody levels, but they also look at T cells and CD8 cells and things that are a better measurement of immunity. And even then, it's going to be, we're pretty sure this is going to last for a while, but we don't really know for sure how long it's going to last, because that's not part of our test. We can't run our test for 5 years to see if it's still running 5 years from now. We can just say it's running, and then we can track these people and see, keep measuring these levels and see if they change in a significant way. That's field testing. That's the real world, and it comes in and bops you in the head. Sometimes you can't test what you want to test, and you need a stand-in. A lot of the early tests in pharmaceuticals are done on animals. Is you want an animal that duplicates as closely as possible the illness or condition that people have. It actually turns out hamsters not only can support COVID, the coronavirus, but they actually get pretty similar symptoms. And mice don't. They'll support the virus, but they don't tend to get sick in a way that's comparable to people. So hamsters for COVID-19 are a better model for that. But they're hamsters, they're not people, which is a weakness when you're running animal tests. And there's 3 basic categories. Animals are good because they're complex systems. You pick an animal, a mouse is another mammal. A lot of its complex systems do duplicate ours. That's the upside. The downside is they're not exactly like us, and their reactions are different in a number of ways. So something that doesn't hurt them sometimes turns out when you start testing in people, it's toxic to people in a way it wasn't toxic in the animals. Downside of that is sometimes something's toxic in animals that wouldn't have been toxic in people, but you never get to find out, because you go, well, it's too toxic to move on to testing with people. You can also test just cultures, particular types of cells that you can get to stay alive in a lab. And they do a lot of cancer testing on this, where you've got cancer cell cultures, and you can throw chemicals at them and see what it does. But they're just cultures. Is that they're not really fully integrated into a living thing. They are exactly the type of cell that you're trying to target. That's the upside. The downside is they don't really necessarily even perform like those cells would in a living system. It's a good preliminary type of modeling, but it's very, very limited. And then you have computer simulations where you build a very complex system based on the information that you know, and get it to do things that, like, run this 50 years into the future, or model the complexities of this cell system and see if it can't tell you something that you didn't already know. They're not really that good at that. They're trying, to come up with artificial intelligence to come up with candidate molecules for new drugs. It turns out they're not really that much better than people using the same information, coming up with the same basic ideas. But in terms of climate simulations, very complex mathematical systems, they can be very, very useful. But they are limited by what we know to put into them. Is that in climate simulations, over the long haul, we don't really know what to tell a computer about how to factor in clouds. Clouds are very ephemeral, they're there and gone. We know that they reflect light, they hold in heat, they're pretty important, but we don't really know how to tell a computer how to factor clouds in for a 50-year span. So we don't necessarily integrate clouds into climate simulations because we don't really know what to tell the computer to do with it. So computer simulations can do a lot of stuff that we can't do, but they are limited by what we can tell them about the system. Sometimes they can figure stuff out, a lot of times they can't. Okay, that's a good place to stop this. We'll continue the design information in the next one.